This research begins in the rural landscapes of Alentejo, southern Portugal. At first glance, the region appears pastoral and calm, but the reality reveals a stark contrast between natural ecologies and extractive systems. In the Mentado area, we found rich biodiversity. Lichens of various species flourished on rocks, trees, and soil. The air felt clean. The ecosystem seemed balanced, with no obvious human intrusion. But just a few kilometers away, the landscape is radically transformed. Massive olive plantations stretch to the horizon. Agricultural machines run endlessly. Pesticides and fertilizers saturate the soil. And nearby, olive oil factories release smoke and residue into the air. Through our fieldwork, we traced this toxic chain, from cultivation to processing. This is not just agriculture. It is a system designed for maximum yield, often at the cost of ecological health. What we observed was not just environmental degradation, but the shrinking of space available for other forms of life. In this landscape shaped by productivity and profit, what remains for the organisms that do not serve this system? This is where lichens appear, not as the focus, but as a symptom. They cling to the margins: stone walls, neglected trees, abandoned infrastructure. They don't just grow, they endure. Their presence reveals an ecological pressure map. Our research emerges from these tensions, asking how we might learn to read the subtle signals of species pushed to the edge. Lichens are complex organisms formed by the symbiotic relationship between fungi and algae. This symbiotic relationship allows lichens to survive in extreme environments, such as rock surfaces, tree bark, and poor soil. The internal structure of a lichen is divided into 5 layers: the cortex, algal zone, medulla, lower cortex, and rhizenes.
The concept of a lichen appears to be simple enough. There's a fungus, which has an incredible capacity for spreading and decomposing, but no means to make its own food. And then there's the algae or cyanobacteria, fully capable of making their own food, but in need of some help getting around on land. The result is called a dual organism, made up of the fungal microbiont and the algal photobiont, that takes on the spreading qualities of the first and the colors of the second. Fungi and algae can meet and work together in ways that actually don't result in a lichen. The distinct structure that we associate with lichens comes down to the thallus, made up of distinct layers that reveal the different combinations of the organisms that make it. The thallus is what allows lichen to survive, keeping the organisms alive in dry conditions so that it can tolerate extremes in humidity and temperature. The shape is generally a function of the fungus, which starts to weave its body into the appropriate layers as soon as it comes upon algae to team up with. At the edge is the cortex, where the fungal cells are thicker and close in contact so they can form a protective structure. And beneath, it keeps a more fibrous and loose mass of hyphae, a scaffolding structure called the medulla that the photobionts layer themselves on, dividing and creating their own algal layer that's large and green and positioned to access the sun. That layer of cells tells us a little bit more about the organisms inside the lichen, though only under the right conditions. A dry lichen is typically gray, but with water, the fungal gray clears up, revealing the color of the photobionts inside the lichen.
A straight road cuts through the fields. On either side, an orderly repetition, endless olive trees. They stand in uniform rows, like a trained regiment. There is no extra sound in the wind, only the rustle of machines and plastic sheeting. This is not a natural landscape. It is land that has been designed, managed, and exploited. Vast, yet lifeless. This land appears prosperous, but it has long lost its original diversity. The bounty is only a facade, the real nutrients have already been drained away. We are not walking forward, we are circling endlessly within a cycle of extraction.
When we looked for lichens, the first thing that caught our eye was their absence. Lichens, which are almost extinct here. But we didn't stop looking. AZPO is an olive oil processing plant, and the black and grey gases constantly emitted from the chimney contain suspended particulate matter, fine dust, nitrogen oxides, and organic chemicals. Around the AZPO olive oil factory, lichen diversity changes with distance. Near the plant, only pollution-tolerant species such as Xanthoria parietina and Caloplaca citrina remain. It sits on the edge of an intensive olive grove and is notable for the variety of brightly colored lichens growing on the surface of the tower, particularly the Xanthoria parietina species. These lichens often possess a certain degree of pollution tolerance and can survive on nutrient-poor surfaces containing nitrogen pollution. Their presence is both a response to the exposed environment of this structure. In Quitos, lichens cover old stone structures surrounded by vast irrigated fields, showing a dominance of bright yellow forms that quietly register the pressures of intensive land use. This is not the result of nature, but of human selection. Lichens change color, grow slowly, or even disappear altogether. This is a slow and continuous expression of them. But no one is there to see or hear it. Just as there is also no one and no government to listen to the local population. Ecological neglect overlaps with social neglect here. During our field trip in Alentejo, we carried out a series of on-site experiments to investigate how environmental stressors influence lichen health. We collected lichen samples from varied environments, ranging from intensive agricultural zones to factory-adjacent sites, for later microscopic observation. To assess particulate pollution, we placed white fabric at human breathing height across different distances from factories, later analyzing the PM10 accumulation on their surfaces. Additionally, we tested the pH levels of both tree bark and the lichen samples to understand how chemical conditions might affect their survival.
São menos de 20 metros que separam este novo olival superintensivo da casa de Gertrudes, onde viveu toda a vida.
Estou tão triste, estou tão triste por causa disto. Eu já me dói tanto a minha cabeça só de pensar nisto. Vão pôr aqui ao pé do quintal.
A plantação teve início há pouco mais de uma semana. O olival entra em plena aldeia de Ervidel, no concelho de Aljustrel. A população está preocupada, principalmente com as...
Um problema na...
Xanthoria parietina, in its natural state without excess nitrogen, appears mostly green. But when factories release nitrogen-rich emissions into the air, these particles are absorbed by the lichen through tiny pores on its surface. The nitrogen stimulates the algae living inside the lichen, speeding up their growth and metabolic activity. As a result, more lutein, a yellow pigment, is produced, gradually shifting the lichen's color from green to a vivid yellow. This silent transformation is not just a change of color, but a visual record of air pollution written on the lichen's surface. Based on the local nitrogen pollution situation, we conducted a visual simulation of the color change in Xanthoria parietina lichen. Based on the color change simulation, we realized that the lichen's response to nitrogen pollution takes a long time to manifest. Therefore, we aim to create a visualized electronic system, an electronic lichen, to help us perceive the lichens' changes within a much shorter time span. When ecology is pushed out of cities, let's bring them back in. Not to mimic nature, but to bring back life that was once banished, in another way. Projection can't replace real growth, but it briefly occupies otherwise neglected surfaces. Stone walls, bare urban facades, the rough skin of tree trunks, acting as a temporary reclamation of space. These projections are not just visual gestures. They carry the political weight of resistance. They challenge the logic of ecological colonization, rejecting human-centered narratives that dictate which forms of life are allowed to remain. In their quiet glow, they become both satire and warning, a reminder of the lives pushed to the margins, and the emptiness left behind when diversity is erased. We are not restoring lichens. Instead, we stand beside them, using light as our shared language, to question who has the right to occupy space, who is excluded, and how architecture might create room for more than just human survival.